Good afternoon, everybody. My name is Matt Brown. I'm the Genomics England Chief Scientific Officer. Thank you very much for joining us at the Genomics England Research Seminar. Just to note that the seminar is recorded. It's available afterwards on our YouTube channel, which should have been in the Outlook invite that you received for this meeting. The seminar today will be 1 hour in length. There are questions and answers after each speaker. Down the bottom of the Zoom window, there's a Q&A feature. So we take questions by you putting them in the Q&A feature, and then we'll address those to the speakers. So rather than putting your hand up and trying to ask a question verbally, can you please put them in the chat? Both speakers have agreed to provide written responses to questions that we don't get through during the seminar itself. So if you ask a question, you will get an answer. So please do address questions in the chat like that. Okay, so going to our speakers, we have two speakers today, Chris Clarkson and Arianna Tucci. I'll start with Chris. Chris is a postdoc in computational biology. Chris and Arianna are both at QMUL. Chris is working in Arianna's laboratory. He has a PhD in epigenetics from University of Essex, and he's working on repeat expansion disorders using Genomics England's dataset and platform. Without further ado, actually, I think we've got one more slide after this just to flag the next seminar. So the next seminar will be the last Tuesday of the month at 2 o'clock, so 26th of November at 2 o'clock. We have two talks, Prima Sanjaya from University of Helsinki and Daniel Green from Mount Sinai School of Medicine in New York, who many of you will remember has recently been on talking about the renewed neurodevelopmental non-coding RNA genetic discoveries, and I think that's an extension of that presentation. So without further ado, I'll pass over to Chris to start his presentation, and just remember questions in the chat, please. Over to you, Chris.
Okay, thank you for having me. Can you hear me okay?
Yes.
Okay, and okay, it just takes a while to set up. Okay, thank you for having me. There's an echo somewhere nearby me, so I'll just really quickly, 1 second, try to find it. Turn it on. Yeah, thank you for having me. As Matt mentioned, I'm in Arianna's lab, and I'm going to be talking about repeat expansion loci and the structural variation that can happen at these loci, and how said variation can affect one's chance of having a repeat expansion. And we also explore it across the prevalence of these structural variations across genetic ancestries when we do this on the 100,000 genomes platform. So I'll give you a bit of background first. Short tandem repeats are a class of genetic variation that are relatively unexplored in genomic sequencing studies. Basically, you have these, I hope you guys can see my mouse. You have these motifs that occur in tandem, and they are generally in non-coding regions, and when they expand aberrantly, they can cause what's known as poly-Q toxicity. In this case, if this tandem repeat was occurring in an exon, CAG encodes glutamine, shortened to Q as a protein, and when they aberrantly expand, poly-Q toxicity ensues, and this is what's known as a repeat expansion. And the resulting phenotypes or symptoms from this are neurological disorders. So there are about 80 different loci, STR loci, across the genome, the human genome that is, that are known to cause repeat expansions or that are associated with neurological disorders due to repeat expansions. So for example, we have Huntington's disease. The gene in question is HTT. The motif is CAG. Generally speaking, CAG polyQ toxicity is a common source of repeat expansion. And as I said, yeah, a handful of loci have been associated with this. So the size in a person, in a given allele of the CAG repeat associates with the age of onset of the disorders that happen. So if you have an expansion of about 60 or above, the onset of the symptoms will start very, very young, whereas they will happen at a much older age if they're lower in size. But a source of variation that's less discussed in this field are the constitution of the motifs within the repeat body. So here I have these interrupting CAA elements interspersed throughout the CAG repeat body. And these are associated with repeat stability. Their presence, rather, is associated with repeat stability. So I think Gao et al. Found when studying SCAT, a type of ataxia or repeat expansion disorder, that pure CAG repeats, those that are uninterrupted, tend to be more unstable, whereas ones where CAA interruptions are present are more stable. So this is a phenomenon that we were interested in. And then, of course, there's another type of variation that can also happen that doesn't happen within the repeat body, but rather at the border of it. And these types of variation can also associate with both the length and chance of having a repeat expansion when at the boundary of the repeat body. Yeah, so having taken you through the basics, I'll show you a bit of the work that we did on the HTT locus, which is associated with Huntington's disease. So the motif in question is, as I mentioned, it's a CAG, a polyglutamine encoding tandem repeat. It occurs in the first exon of the HTT gene. And our colleagues did work on this locus, whereby they characterized the structural variation that can happen across this repeat. So at first you have the polyglutamine termed Q1 locus, the Q1 repeat in this locus, and subsequently afterwards, the less explored, you have Q2, which is a CAG repeat, and its presence strongly associates with the length of the Q1 repeat. So, for example, if you have a duplication of the CAG, it associates with a shortened Q1 repeat, versus its total absence associates with a longer Q1 repeat size. And then, of course, subsequently, there's a proline encoding locus or repeat that also associates when it's missing. That associates with a longer Q1 repeat. So our colleagues did this using a form of sequencing called MySeq, which was tailored specifically to this locus. Now, we, on the 100,000 genomes platform, we wanted to explore the structural variation at scale. They did it in a relatively small sample size, but we wanted to do it using whole genome sequencing, which is short read data. And we also wanted to do it across many, many more samples. So I'll take you through how we did that. So when you're using whole genome sequencing data, the typical tool that you would use to study tandem repeats is known as, it's a bioinformatic tool called Expansion Hunter, and it takes BAM file read data, and it will also take a prerequisite list of motifs that will be present or not in the BAM data, and it will count the presence of the tandem repeats that it sees in the read data. And there is, however, a limitation to this, because here we have two alleles. There's an allele where one CAG is present and then 5 are present in the other one, but we don't know how these vary in relation to the presence of this one. So that is to say, these tandem repeats and their presence is not phased. So here we can't, for example, say whether the CAG times 1 goes with a CAA CAG times 2, in which case CAG times 5 would go with the CAA CAG times 1, and or vice versa. We can't tell which of these scenarios applies. And that's a problem, because we then subsequently wouldn't be able to study how they vary in relation to one another and correlate with one another. So to handle this problem, initially we wanted to study the read data manually by just looking at the actual read data. And you can see here, pretty striking immediately, that so we have the Q1 repeats, and subsequently there's a loss of the Q2. And you can see that this repeat is longer than in the case of the allele in the same sample where a canonical repeat structure is present. That is a single Q2 preceded by a Q1 of a relatively short repeat size. So it's good to know that you can do it manually, but that's not scalable. So we wanted to do this for many different genomes, as I said, across the GEL platform. So we invented a tool called Repeat Crawler, which is another bioinformatic tool that complements the data from, or the output from, Expansion Hunter, and works similarly, except it not only counts the presence of each repeat element that's input, but it also phases them. So here we have a host of reads that are indicating a particular repeat structure, but it allows us to separate different alleles out according to, number one, their one repeat size, but subsequently it also allows us to actually assign a repeat structure to said repeat size. And in that case, we can actually start to study at scale, as I said, these repeat structures and their effect on one's chance of having an expanded Q1 repeat in the HTT locus. So we were pretty happy with the fact that we were able to reproduce our colleague's previous findings, in that the duplication of Q2 strongly associates with a shortened Q1 repeat size relative to all other repeat structures, and Q2 loss, as was expected, associates with an overall generally larger repeat size, Q1 repeat size. And then the loss of both Q2 and P1 strikingly has a much larger repeat size generally than all other repeat structure cases. And of course, because it's Genomics England, we were able to avail of genetic ancestry data. And yeah, so we can see that there are substantial differences in terms of the prevalence of different repeat structures across these different genetic ancestries. So it'll be interesting. It was nice that we could reproduce our colleagues' results, but it'll be nice going forward as more diverse cohorts and populations are sequenced to be able to take the differing presence of different repeat structures into account while considering a person's chance of having a repeat expansion. So yeah, that was work that we published just recently in Nature, work done by Christina, our previous lab member, and I was able to complement her work with Repeat Crawler. So having done that, we then wanted to expand and study other types of repeat structure that could affect a person's chance of having a repeat expansion. So FAP11 is a gene that was associated with ataxia in a small cohort of Chinese people in northern China. And it presented an opportunity to us to study repeat structure, because I'll just take you through the actual structure of the locus. So there was a family in China where ataxia was quite prevalent, and in people who didn't have ataxia, the structure of this locus went a tandem repeat of CAG followed by a single CAA, tandem repeat of CAG, and so on. And there were 4 to 5 CAA interruptions in cases where the disease was not present. However, when it was present, there were only 3 CAA repeats with an expanded or a relatively expanded 3' CAG repeat, glutamine repeat. So that presented us with an opportunity or a challenge in that we wanted to see, given that this was found to be extremely rare, because it could only be found in China, we wanted to then search for it on the gel platform, number one. So we found a handful of European people who had a sufficient repeat size to suffer from ataxia or to be characterized with ataxia, according to this paper and the threshold of repeats. However, this person with a repeat size of 47 didn't actually have ataxia, and we couldn't find anyone with a sufficient repeat size who also had ataxia on the gel platform. So we wanted to be able to try to explain this. Now, I'll mention that the people who had the sufficient repeat size that we could find had 6 to 7 CAA interruptions, which is longer or more interruptions than that of the people who were found to have ataxia in the Chinese cohort. So again, this presented another opportunity to study with repeat crawler. It was perfect in terms of its application to this problem. So we were able to ascertain in each allele that in a large population the number of repeats, the number of interruptions in the repeat body in FAP11, and you can see that the overall repeat size correlates strongly with the number of CAA interruptions, which was something that we hadn't previously observed in any other repeat or had observed in any previous literature. And furthermore, we were also able to ascertain the prevalence of these repeat structures in different genetic ancestries, and the one with 3 interruptions, that being the one that associated with ataxia, was most prevalent in African and South Asian genetic ancestries, those being relatively under-sequenced populations. And furthermore, the longest pure CAG repeats that we could find in this population was 20, which is nowhere near that seen in the ataxia patients in the cohort where this was originally discovered. This expansion was originally discovered. And finally, we were also able to measure the length of the in-body repeats, so the pure CAG repeats, and we took the 3' CAG body length in repeats with 3 interruptions, and we studied them across different genetic ancestries. And as you can see, there are significant differences in the length of this repeat element across the different ancestries. So it just goes to show that different genetic ancestries will have different prevalences of repeat structures, and subsequently of one's chance of having repeat expansion. And we were happy to be able to make this clear and to explore this, and we'll be able to take it further in subsequent studies and study things that previously studied, potentially even novel repeats rather than just known repeat expansion loci. So I hope I'm not too far underneath. Okay, I've got 4 minutes left, but yeah, so thank you to Arianna, my boss, and the lab in general, because it was a really interesting study that we were able to use. So repeat crawler has allowed us to characterize sub-repeat elements, and one's chance as a function of structural variation in a repeat expansion locus, one's chance of having an expanded repeat. And we also showed that certain repeat structures associate with genetic ancestries. And in your feedback, I'd love any advice that people could give in terms of how we could actually improve the pipeline repeat crawler, because right now it just relies on the order in which one specifies the repeat elements. And I think it could potentially be even more improved if it were possible to use something like a hidden Markov model, which is agnostic to the order. It doesn't need there to be a specific order, and something like that would be really, really useful for us to discover potentially even novel repeat structures that we haven't seen before. So thank you. Thank you for having me. And if you'd like to ask me questions, I'd be happy to help.
Okay, so we'll go to questions now. So if people can, again, if you can put questions in the Q&A that's down the bottom of the Zoom screen, that would be perfect. And in between times, I'm going to ask a bunch of completely naive questions. So Chris, I'm just, it's apparent that most or all of the repeat expansion disorders, certainly the ones that I know of, neurological or neuromuscular. Wondering
Yes.
if you can give us a hint as to why you think that might be. Why are the cells involved in those particular cell types particularly susceptible to these conditions?
Yeah, it's a good question. I probably should have put something in the presentation about that, but Arianna will know much better than me, and she'll be able to really answer that thoroughly. As far as I'm aware, the polyQ toxicity, it results in neurological disorders, particularly because the region of the brain, the cerebellum, and I don't think it's known why, but it's particularly unstable in the cerebellum, and hence, for example, when you get ataxia, it's a function of these expansions being in the cerebellum, and so the symptoms that manifest are because of your cerebellum is responsible for your balance, and ataxia is known to affect one's balance. As to the molecular basis as to why it actually happens that way in terms of polyQ toxicity, I think people are still investigating this, and it's not fully explored, but Arianna will definitely be able to give you a much more thorough answer on that.
Okay, well, I might come back to her and also ask about
Sure.
why some of these diseases are progressive versus others just being late onset. So I think that's really interesting. But a few questions from the chat. So firstly, Alice Davidson, is repeat crawler currently available freely?
Yes, it is. Yeah, I should have actually put a link to our GitHub,
Okay.
and I can email that around if that's possible. Sorry, that's my time.
Why don't you put it in the chat afterwards, and that way
Sure,
everybody.
we'll do. We'll do.
Okay. Shrivashini Srinivasan, apologies if I mispronounced your name, from QMUL, wants to compliment you on your presentation, and wonders if the analysis that you've done can be done similarly with copy number variants.
Okay. Yeah, it's an interesting question. I think copy number variants would be a bridge too far for us, because they're so large. So copy number variants tend to be kilobases long, whereas our loci that we tend to study, we rely on short read data. And having short read data, it's like having a measuring pencil for measuring from here to the end of the hallway. It's not quite enough. So we rely on reads being placed relative to one another and being aligned fairly accurately. So we have to pull all sorts of tricks in order to characterize the structure of repeat expansion loci um, when studying expanded repeats or expanded STR loci. So in terms of copy number variance, I don't think it could quite do the job, but it'd be interesting to see if there are elements at the border of a CNV that correlate with the number of CNV elements. I think it's an interesting question and could be something that could be studied in the future. I don't think our tool is quite applicable right now to this sort of that sort of variation. But thank you for the question.
Okay, thank you. So then Anonymous, generally a very good questioner, has asked a question about the ancestry-specific nature of repeats and about whether those have been replicated. I'm going to add to that and just ask, why do you think there is an ancestry bias? Are these common founder variants? And if so,
Yeah.
what's the selection advantage which enables them to be maintained?
Right. So we haven't speculated on that yet. We've published this work, but we don't fully know because we don't have. I mean, it's very clear that certain repeat structures are a lot more prevalent in certain genetic ancestries than others. But I don't think we have enough data to be able to say. I think we would want to study actual instability events whereby a repeat expansion occurs, and we would know that by observing a proband in two parents, whereby the proband has a repeat expansion that's not present in either of the parents, and being able to study, and we could definitely do this with a repeat caller. We could study the changes in, say, the repeat motif or the number of interruptions that are present in a given repeat body.
Do you know if there are
To,
actually, if they have linkage disequilibrium with variants around them?
say again.
Do you know if they have linkage disequilibrium with variants surrounding them?
So we know from the Huntington's locus that other people are studying this, and we know that certain repeat structures like a duplication of Q2 will be in LD with a relatively short 7 to 11 repeats of the Q1 locus. We know they're in LD with each other, but in terms of distal variants, we wouldn't know. Or even maybe semi-distal variants that aren't immediately after the repeat body. I don't think anyone has studied that.
I just want to move on to one more question from Andrew Wilkie, another person who asks great questions, who questions about triplet repeats generating lots of sequencing errors through slippage.
Yeah.
And how do you manage these bioinformatically? And are you always able to identify two definite alleles? And then asks about whether true mosaicism occurs in some individuals.
Yeah, it's a good question. So our stringency threshold for knowing or for accepting an allele is that we have to have at least two spanning reads before accepting that a given allele is of a particular repeat. We do get mosaicism, but we only accept from repeat crawler or calls that agree with Expansion Hunter, and Expansion Hunter avails you of a bunch of different metrics whereby you can study the QC or study the quality of the reads in question. Just having studied reads manually, yeah, so we have to accept a certain, it depends on the locus, but we have to accept a certain number of reads and their quality. And any reads that aren't of a good quality are actually discarded by Expansion Hunter. So there's a range of other tools that also enable you to assess the quality of a given region. As to mosaicism, if there's a relatively small number of reads indicating something that's different from the two most supported, most robust repeat structures or repeat sizes, then we tend to discard that. You're right in that there's sequencing error. As of right now, we're limiting this analysis to regions that can be spanned by 150 base pairs in terms of the read size that you get in whole genome sequencing. So we can't be as prone to false positives due to those kind of sequencing errors. So thank you.
Chris, thank you very much for that. Perhaps if you could just put the address for.
Will do. Actually, I'll do that now. Thank you.
Is it being published? Because putting in the reporting publication might be helpful for people too.
Yep, will do, of course. Okay, thank you.
Okay, thanks very much, Chris. So now we'll pass to Dr. Arianna Tucci, who's a clinical reader in genomic medicine at Queen Mary and an honorary consultant in clinical genetics. Her primary research focus is neurogenetics, and she's particularly interested in using emerging technologies to identify improved diagnostic methods for these different approaches. So her talk today is about following on the theme, increased frequency of repeat expansion mutations in the general population. Over to you, Arianna.
So thank you very much for inviting me. Let me share the screen. Okay. Yeah, really excited to be here today to talk about a work that really started a few years ago when I was working as a clinical fellow in the rare disease team at Genomics England and studying how whole genome sequencing could detect short tandem repeats. So you just heard from Chris about the repeat expansion diseases. I just touch on the fact that they are a heterogeneous group of disorders, but they are caused by the same mutational mechanisms. They are caused by a repeat expansion mutation. So the repeat expansion mutation involves naturally occurring short DNA sequences. So in the human genomes, there are many of these, but they tend to be short. The repeat expansion mutation causes the number of repeats within this repetitive region to increase beyond a certain pathological threshold, and this is what causes disease. Okay, this is a non-exhaustive list of repeat expansion diseases. The list is growing thanks to the technological advancement of the last few years. But this is really, again, just to give you an overview of what they are. They are mainly late onset neurodegenerative diseases. They mainly involve the nervous systems. There are some notable exceptions like the Fuchs corneal dystrophy, which is not a repeat that is studied here. As I said, mainly involve the nervous system. They present mainly at late onset neurodegenerative diseases, but they are also one of the most common cause of non-syndromic intellectual disability, like Fragile X syndrome. Here they are sorted by the most common, but the most prevalent disease to the least one. So the most common ones are Fragile X, C9orf, frontotemporal dementia or ALS, myotonic dystrophy and Huntington disease, and the least frequent ones are the spinocerebellar ataxia. So we are looking at, if we look at the known disease prevalence which is described as the number of people affected by the disease by 100,000 people. Like, for example, the Fragile X syndrome affect 14 to 20 people out of 100,000, and the spinocerebellar ataxias, which are a lot rarer, 1 to 2 people out of 100,000. Some of the repeat expansion diseases are population specific, like ATN1, which is mostly been described in populations from the Japanese population, or Huntington disease like 2, which is mainly been described in Africans. So one of the hallmark of repeat expansion diseases is that they are both clinically and genetically heterogeneous. So clinically, the same repeat expansions can present with different phenotypes, as you heard from Chris. And also genetically, different a repeat expansion can cause the same phenotype, for example, the spinocerebellar ataxia. Again, this is just a very well-known example of clinical heterogeneity, the myotonic dystrophy, which is caused by a CTG repeat expansion in the 3' end of the DMPK gene. And there is a clear relationship between the size of the repeat and the age of onset and the phenotype. Small repeats, between 50 and 100 repeats, cause a late onset mild disease, which mainly present as cataract, whilst large repeat expansions involving many thousand repeat units cause the congenital form, which is a multi-system disorder, which involves early death. So same repeat expansion, different repeat, massively different phenotype. So the heterogeneous clinical presentations limits what we know about the epidemiology of these diseases. So epidemiological studies in repeat expansion disease are challenging. Traditionally, epidemiological studies are based on the clinical observation. So patients are identified when they are diagnosed with the disease. And then the number of people that have diseases are counted out of a denominator, which could be a local population, for example. So epidemiological studies require the clinical observation and a diagnosis. And this means that some mild asymptomatic carriers may not be tested, and that sometimes also because, as I said, some of these repeat expansions have been traditionally described only in certain populations, some populations are not diagnosed for these diseases. So there is a widespread feeling that there is an ascertainment bias and that there is an underestimation of the true prevalence of these repeat expansion diseases. Another way to look at the epidemiology of genetic diseases in general is to use genetic testing. So traditionally, genetic testing for repeat expansion diseases relied on target single gene or small gene panels, PCR-based methods. Sorry, I put the laser on. Yeah. So traditional genetic testing relied on PCR-based methods, which limit the scale in which repeats can be tested. The largest study to date on the prevalence of repeat expansions based on genetic testing was published by Aziz in 2019 and involved, as I said, PCR-based of the CAG repeats for 16,000 individuals that were collected over a period of 15 years. So the limit, as I said, on time and scale of what can be done. So we thought, can we use whole genome sequencing and the large data repository of the whole genome sequencing to measure the frequency of these repeat expansions? Of course, in order to answer that question, can we do it using whole genome sequencing? We need to have a tool that works. And in the last few years, a number of bioinformatics tools have been developed to genotype repeats from whole genome sequencing. We used a tool called Expansion Hunter, but there is a large number of these tools. And in principle, they all worked in the same way. What they do is to scan a BAM file, whole genome sequencing file that comprises short DNA reads, to identify reads that either completely contain the repeat, here highlighted in red, or partially contain the repeat, like these reads. And what this means is that for repeats that are shorter than the read length, we can pretty accurately estimate the size, whilst for repeats that are larger than the read length, we can compute the repeat size with confidence interval. So, as I said, number of tools developed to detect repeat expansions from whole genome sequencing. However, to answer our question, which is what is the frequency of repeat expansion disease carriers, we need these tools to be accurate. And we have previously shown that using Expansion Hunter in the Genomics England dataset, that a subset of repeat expansion diseases can be detected from whole genome sequencing accurately and actually with high specificity and sensitivity. And so this is really reassuring because it tells us that the tool and that we are using to answer the question is accurate. Of course, there is a limitation to what whole genome sequencing can do. in terms of predicting the size of repeat expansion. And the limit, of course, is the sequencing read length. So this plot shows on the y-axis different repeat expansion diseases, for example, Huntington's, spinocerebellar, Friedreich ataxia, SBMA, myotonic dystrophy, and so on. And on the x-axis, the repeat length. In different colors are different repeat categories. So normal repeats are in blue, intermediate in yellow, and red are the full expansion of pathogenic repeats. And this is the sequencing read length. So anything smaller than this sequencing read length will be sized accurately. For example, all repeats that are normal, intermediate, and some of the expansion in Huntington's, whilst anything larger than the sequencing read length cannot be accurately sized. For example, the repeat that causes Fragile X syndrome, the technology cannot distinguish between an intermediate or pre-mutation allele and a full mutation. So we went on to say, okay, we can use this data set to study the frequency of repeat expansions, but given that some of these data sets and some of these repeats are also population specific, can we look at how different repeat expansions are distributed based on the specific genetic ancestry? And to do this, we use 3 different cohorts. So the first one is the 100,000 Genomes Project cohort. And of course, I guess all of the people on this audience know about the 100,000 Genomes Project, a UK research project, which was set a few years ago to test the utility of whole genome sequencing for patients with rare diseases and cancer. Then we use the US counterpart for late onset cardiovascular and respiratory diseases, which is the TopMed dataset, nearly 50,000 genomes, and that the 100,000 Genomes Project data, which of course comprises a much smaller number of genomes, but the representation of different population is more homogeneous. And the reason why we use these data sets is, one, because that was available a few years ago. They were all sequenced using the same technology, whole genome sequencing 150 base pair reads with Illumina. They all had deep phenotyping data, and some, especially the TopMed, had different diverse ancestry backgrounds. So this is the overview of the studies. So we merged the TopMed and the 100,000 Genomes Project cohort. So this cohort is mainly comprised of nearly about 82,000 genomes, and the 1,000 Genomes Project 2,500. We selected unrelated genomes. From both the Genomics England and TopMed cohort, we excluded genomes from patients that were recruited with the neurological disorder. And the reason why we did that was to avoid overestimating the number of people that carried a repeat expansion, because they were in the data set for the specific, for the very reason that they presented with the repeat expansion disease. We then went on to assign ancestry based on the 1,000 Genomes Project projections and PCR analysis, and this is the cohort divided by genetic ancestry. Of course, is mainly European, nearly 60,000 European genomes, 12,000 Africans, 5,000 Americans, and a smaller number of South Asian and East Asians. If we look at the 1,000 Genomes Project cohort, as I said, this is a more homogeneous in terms of population. When it's broken down by population, it's more homogeneous, so about 500 samples from each genetic ancestry. We went on to use Expansion Hunter version 3, and we visually inspected all repeats that were larger than the sequencing read length. So to study the carrier frequency of repeat expansion, we selected the genes where we previously shown that whole genome sequencing can accurately distinguish between full mutation and pre-mutations. So, for example, the CAG repeats or the polyglutamine diseases. And we also included larger repeat expansions like the C9orf, myotonic dystrophy, and even RFC1, which is which is caused by a very large repeat expansion. And for this, we work with someone who had developed a tool specifically to call this repeat. We also extended our analysis to repeats where we know we can't distinguish between pre-mutation and full mutation but where we were interested in looking at the distribution of alleles in different populations. So this is what we found. The first thing that we looked at is the mutation frequency. So here is a version of the table that you've seen this before. Again, repeat expansion diseases sorted from the most frequent to the least. But I've added a column here, which is the mutation frequency. So we've confirmed the fact that the C9orf, the myotonic dystrophy, the Huntington's are the most common repeat expansion diseases. However, when you look at the difference between the mutation frequency, as we've detected, and the non-disease prevalence, the difference is quite remarkable. This is across most of the repeat expansion diseases that we studied, as I said, myotonic dystrophy, Huntington disease, C9orf, but also for the spinocerebellar ataxia. I think this was really an unexpected finding for us. Whilst for some of the spinocerebellar ataxias, we confirmed they were specific to some populations. So what I'm showing here is the mutation frequency compared to the non-prevalence. However, we know that the mutation frequency doesn't equal the number of people that go on to develop a disease, because repeat expansion diseases are late onset disorders. Mainly, they are characterized by reduced survival, and some have very well, or not very well, but defined age-related penetrance range. So we tried to use the genetic data to model, knowing the number of people in the population that carry the disease expansion, can we model the number of people that we expect with the diseases? And how did we do that? We did it for, the prediction is based in the UK population, because we have, thanks to the Office of National Statistics, we know exactly how many people there are in the general population in UK divided by age. And this is really simple maths, is a series of multiplications and subtractions. So we take the UK population by age. So sorry, here on the top, the top panel shows the workflow of how the disease modeling works. And the bottom plot shows an example. This is C9orf. So we take the UK population count by age. Each count by age is multiplied by the carrier frequency. Here, for example, C9, 1 in 800 people. And then we took either from literature or from patient registries, such as the EuroSCA for the spinocerebellar ataxia, or the myotonic dystrophy registry, the age of onset distribution. So again, we multiply that by the carrier frequency. For some of these diseases, as I said, we know that they have reduced penetrance. So we subtract the people every year that die, and we come up with the prevalence estimate, which we compare. So prevalence estimate by age, here on the x-axis, the number of people which we expect to find with the disease. And then we compare to what we know from the literature based on observing the disease, here in red. And again, what do we find? That across the board, our estimate of people that we expect to present with the repeat expansion diseases is 2 to 3 times, in extreme cases, even 9 times higher than what is reported based on observing the disease in the general population. Here you see the example for myotonic dystrophy, the spinocerebellar ataxias, and Huntington disease. For Huntington disease, we did our modeling based on 40 CAG repeats, because a large number of people that we've detected in the general population with the expansion carried 40 CAG repeat, which is the smallest repeat size that is enough to cause disease. So. And again, we found 2 to 3 times higher predicted prevalence of disease. So what does this mean? I think, yeah, this plot really, this is an image that shows that there is a discrepancy between the number of people that are observed with the disease versus the number of people that are predicted to develop a repeat expansion diseases. I have to note that a number of people that we found with carrying the repeat expansions have a repeat expansion which is pathogenic but small. Given the fact that we know there is an inverse relationship between the repeat size and the age of onset and the severity of the disease, one is left to wonder whether either we are underdiagnosing some of these repeat expansion diseases that are caused by small pathogenic mutation that present with a milder phenotype and are later onset, or whether actually these smaller pathogenic expansion do have some markedly reduced penetrance. The third option is that they present with the different phenotype that is not very well described to date, i.e. variable expressivity. Some people that we've detected with the large expansions, according to the data that was available to us, did not express the disease. And there are several reasons. I mean, these are hypotheses which we need to test. Why do these people do not express the disease? Are there some modifiers of disease that prevent these people from developing it? Or actually these are technical fault, i.e. the clinical data has not been captured either by the 100,000 Genomes Project or TopMed, and a very small number of people did carry large expansion that didn't reach the age of onset yet. So the second part of this talk is about looking at the relative frequencies of repeat expansions in different populations. And why we're looking at this, because as I said at the beginning, some of these repeat expansions have been described in specific populations. So to answer this question, what we did is to compute the number of carriers that carry pathogenic alleles, so either in the full mutation or in the reduced penetrance range. And then we divided by the superpopulation. So each bar here represents a superpopulation, and each color represents a proportion of different repeat expansions. So if we look at this and we split the repeat expansion diseases by autosomal recessive as dominant, as expected, the large number of people that carry pathogenic repeat expansions are the ones that carry repeat expansions for those diseases that are autosomal recessive. So RFC1, which causes canvas, and FXN. For RFC1, this is by far, of course, the most frequent one, with a carrier frequency of 1, I think, in 12 in the European populations. FXN is to follow, and it's present in most populations except the East Asians. When we look at autosomal dominant repeat expansion diseases, we start noting population differences in the relative frequency of these repeat expansion diseases. And the most different one, as expected, based on what we know about the history of human population, is these East Asians. East Asians carrying a large fraction of pathogenic alleles in TBP, in Notch2, in DRPLA. These are repeat expansion diseases that are traditionally being described only in these Asian populations. However, for other repeats, like the androgen receptor here in gray, SCA1 here in black, ataxin-1, ataxin-2, and even C9-OFF, which are typically described most in European population, they are present across all ancestries. So the last thing that I want to show you here is we did ask whether the different epidemiology of repeat expansion diseases may be explained by the different distribution of repeat lengths in different populations. And in order to understand why we're asking this question, we have to think about the repeat expansion mutation first. So the repeat expansion mutation is a gradual process where large pathogenic repeats normally are thought to arise from shorter intermediate range repeats. These are repeats that are not enough to cause the disease, but they acquire the somatic and the germline instability, and they are more likely to expand in the full pathogenic range. So we did wonder and ask ourselves whether, if we look at the allele frequencies versus the repeat size, this is an example for the myotonic dystrophy, and we look at the different population, at this plot, but a different population, will we find different percentage of people that carry alleles in the intermediate range, which indicates the fact that in some population we will find more or less people with that repeat expansion. And this is exactly what we found. So the myotonic dystrophy here, it's much less commonly observed in the East Asian population. And as you can see here, they have a much shorter number of people with intermediate repeat lengths. We went on to confirm the fact that there is indeed a relationship and a correlation between the frequency of intermediate alleles and the frequency of pathogenic alleles in different populations. This is an example you can see here, the Huntington disease. Each shape, it's a different population. Each population has different proportion of intermediate alleles, which is maybe the reason that underlies the fact that we observe the disease less frequently in some of these populations, for example, in Africans. So I don't want to reiterate what I've said, but just to say the conclusion from this work, I think there are really two take-home message. One is that when we start, I think traditionally we've always known about repeat expansion diseases and generally about rare genetic diseases by looking at families that present in the hospital. And I think actually when we start looking and moving away from families and we look in the general population, we start seeing a different picture. So a lot more that many more people than expected that carry these genetic defects. And whether this is due to the fact that they are underdiagnosed, for example, the small pathogenic repeats, or they have reduced penetrance or variable expressivity, I think this is a question that will need to be addressed using, for example, different data sets. For example, the UK biobank that has longitudinal data and very rich phenotypic data for the older generations. We also shown that many repeat expansions are represented in broad genetic ancestries, for example, Huntington, androgen receptors, and the SCAs, and that the different distribution of intermediate alleles may underlie and may be the cause of observing different proportion of repeat expansion disease people. Of course, there are some limitations to these studies, and the main one is the fact that there are over 50 repeat expansion diseases, but some of them cannot be really studied by whole genome sequencing. For example, the FGF14, this is recently described cause of late onset spinocerebellar ataxia, and I think it's one of the most common cause of late onset cerebellar ataxia, and some of the myoclonic epilepsies as well. As I said, we have limited data on penetrance, and our data set, of course, is still very much eocentric, and now we're moving towards using genomes from more diverse ancestry to understand at the molecular level what happens in different ancestries. So I would like to thank all the people in my group, and specifically Christina, who has been driving this work, starting at Genomics England and now working with me, and then the Institute of Neurology, Andrew Sharp with his group at Mount Sinai, and of course the patients and the families that are part of the Genomics England and the TopMed dataset, and special thanks to the funders as well, UKRI and Bart's Charity. And thank you very much. Wow.
Thanks, Arianna. We have actually hit the top of the hour, but so for those of you who need to move off, please feel free to. Arianna, are you okay to stay on for a few minutes to answer some questions?
Yeah, sure.
Okay. All right. So there are some questions about basically confirmation of data. Alastair Penumenta, who I know works with our RNA sequencing data in Marfan's disease, has asked about RNA sequencing data to validate the repeat structure. And can I add to that, you're aware, of course, that there's a lot of long-read sequence data in the NGRL as well, particularly on neurological disorders. Have you looked at that either to validate or to do any research with?
So question about RNA sequencing. The question about RNA sequencing is to validate whether not, I mean, we did look at the RNA sequencing, but rather to look at whether the repeat expansion has an effect on the expression based on the data that we had, not to validate the length of the repeat. I don't think that work we had done in our previous work that was published in the Lancet paper, where we were able to show that there is a nearly, perfect agreement between whole genome sequencing repeat length and PCR length when we look at repeats that are shorter than the sequencing read length, which is 150 base pairs. The second question about the long reads, I am aware that Genomics England is generating a lot of long read sequencing data, and I'm looking forward to that data set to be able to analyze these repeats, which we couldn't do.
Right, so we don't have time to go through the question I was going to ask about epigenetic change, but there's a question from John Hardy about the potential of having a repeat expansion in one allele and a second pathogenic variant in a second gene. Do you have examples of that? And would that, if so, did it change the phenotype?
We did look limited, I have to say. This is a really good point. And I think we did look for some of these repeats, and we couldn't find a straightforward answer. But that was a quick and dirty job. We didn't do it systematically, but that's definitely something on the table. Also because some of these repeat expansions, for example, the one that we were recently involved in AFF3, a new cause of intellectual disability, it is also caused by single nucleotide variants. So it is very likely that there are modifier factors outside but close to the repeat.
Yeah. Okay, well, with that, I am going to draw it to a close because we are past the hour. But thank you very much both to Chris and to Arianna for really fascinating presentations. I'd encourage you to keep looking at the NJRL because there are several thousand long-read sequence samples in that, 7,500 RNA sequencing data sets, and much of which is on neurological disorders. And everybody who's on the call, thank you for your questions, and I look forward to seeing you again on November the 26th at 2 o'clock. Thank you.
Thank you very much. Thank you for having us.